Welcome to our channel. In today's video, we are exploring the role of DMSO in PCR. PCR or polymerase chain reaction is a powerful technique that allows us to amplify specific DNA sequences. But what is the role of DMSO in this process? To find out the answer, stay tuned at the end of the video. So let's get started. DMSO, which stands for dimethyl sulfoxide, has trigonal pyramidal symmetry. Chemically, it is a 2-carbon sulfoxide having 2 methyl groups. It is a polar solvent which frequently used in PCR reaction. It plays a crucial role in enhancing the efficiency and specificity of the PCR amplification process. In medical field, DMSO also used as a cryoprotectant. Anyway, let's talk about how DMSO does work in PCR. During PCR, the DNA template strands are denatured or separated to allow primer binding and subsequent DNA synthesis. However, some DNA sequences can be challenging to denature due to high GC content. So there are two common problems with high GC content DNA in PCR amplification reaction. First, G and C contains triple hydrogen bonds, which require a higher temperature to break them. Second, more GC-rich templates form many secondary structures in the DNA template, and these secondary structures are hard to open and amplify, so we can't get good amplified product. There might be non-target amplification, primer-dimer, or unwanted amplifications. Now first understand why we need to add DMSO in the PCR reaction. So with a high GC content, targeted DNA requires a high melting temperature, and that might prevent accurate primer binding to the target DNA and also damages the template too. When we add DMSO, the DMSO binds with the cytosine base of the template and makes some conformational changes. This causes cytosine to be more heat labile and eventually drops the overall melting temperature for primers. That means DMSO disrupts the hydrogen bonding within the DNA molecule, making it easier to denature and separate the DNA strands. This allows the primer to access the target sequences more effectively. But not at all. DMSO also helps improve the specificity and efficiency of PCR amplification. Now secondly, higher GC content produces a hairpin-like secondary structure, and these structures stabilizes the single-stranded DNA. Such hairpins are hard to open and fail our reaction by not providing a complementary site for primers. DMSO binds with the DNA and prevents reannealing of denatured DNA. Thereby, only primers get white space to accurately bind at their complementary locations. Now let's compare PCR with or without DMSO. PCR reaction without DMSO may struggle to denature complex DNA structures, resulting in incomplete amplification or false negative results. In contrast, the inclusion of DMSO improves denaturation, primer binding, and overall efficiency of the PCR reaction. DMSO enhances DNA denaturation and overcomes secondary structures, improves primer binding and specificity, and increases the overall efficiency and reliability of the PCR reaction. So that's the major role of the DMSO in PCR. But it's important to note that the optimal concentration of DMSO may vary depending on the specific PCR reaction and DNA template being used. So we should carefully optimize the DMSO concentration to achieve the best results. So this is the end of the video. Thank you for joining us. Don't forget to like and subscribe to my channel for more exciting science content. Stay curious and keep exploring the wonders of genetic research. Until next time, take care. Bye.